Thanks for joining us for this symposium. I'm Bill Rapisi, President and CEO of the Lymphatic Education Research Network. LE&RN's mission is to fight lymphatic diseases through education, research, and advocacy. In order to win a fight, you first have to join it. So we ask, please become a supporting member of LE&RN at lymphaticnetwork.org. And we hope you enjoy today's symposium.
Hello, everyone. I'm Jimmy Moore. I'll be presenting today's symposium. You can see the title here, and I'm very pleased and honored to be invited by LE&RN to do this. I've actually given one of these before that focused mainly on my research. And today I'll be talking about some of the devices that we've been developing that hopefully will help in the fight against lymphedema. I'd like to very proudly acknowledge all of the fantastic support from the sponsors of this series. And without sponsorship like this from these wonderful companies, certainly LE&RN would not be able to do the wonderfully successful advocacy and other research support work that it does. So thanks very much to all the sponsors. There's the standard disclaimer that you get used to seeing at this symposium and perhaps other similar ones. So I'm talking to you from Imperial College in London, and yes, it's raining here, and it is evening time, and but it's really fun to do these symposia. I've been an audience member at many of these events, and they're all informative and wonderful, and I hope I can live up to that standard. So here what you see is a sort of a laundry list of research that I do in my lab, and you can see most of it focuses on various aspects of the lymphatic system. We also are involved in translating the fruits of our research out into the community to try and achieve clinical impact with what we find in our research. So that's some of the theme of what I'm going to be talking about today. So we do some experiments in my lab. We do some computational modeling in my lab. You'll see examples of both of those today. And then sort of the third leg of what we do is device development. So we try and learn from the results of our research to design devices or technologies or strategies that can be that can assist patients and clinicians in improving their lives. So in terms of funding and declarations, you see here I've received funding in the projects that I'm going to be talking about today from the NIH in the United States, from the NIHR in the UK, and from our own university's Confidence in Concept Fund, which is aimed at helping some of these early stage technologies get to the next stages of de-risking. So I will declare that I am the inventor of the technology that I'm going to be talking about mainly this evening. And I'm a founder and shareholder of the company Lymphomotus that is aiming to commercialize this technology. So most of you are aware of lymphedema, but we'd like to summarize sort of the magnitude of the problem in this slide. 20%, and that's an approximate number, I'll come back to that later, of breast cancer patients undergoing treatment develop BCRL or breast cancer-related lymphedema, generally considered incurable, and really debilitating. So not only is there limits in physical function, but there would be social dysfunction, a lot of depression associated with this. So it's not a nice condition, and we should do what we can to help people deal with it. So just trying to get the laser pointer up here. So there are several challenges involved in not only treating or trying to treat the disease, but even just diagnosing it. So it needs constant follow-up for a number of years after surgery in order to determine if the patient's going to develop BCRL. Not all clinicians are as aware as they should be about lymphedema. And it's very costly once it does develop. And so that's why us and several other groups have focused not only trying to treat BCRL, but in fact, what I'm going to focus on today is more about prevention. So BCRL prevention, we think, can address many of these shortcomings in healthcare systems and offer an improved quality of life to breast cancer survivors who, let's face it, they've been through enough already without getting a swollen arm. So some of the perspectives that we've gained by interfacing with our various stakeholders, which is an important part of the technology transfer process, so are listed in this slide. So we've engaged with a number of patients who have lymphedema, and you can see this would be representative of what you can find on LE&RN's website. And I'll just point out this last one here, which is not something that sort of I hadn't heard before. And so this particular patient uses a pneumatic pump to help her deal with her lymphedema. And every time she switches it on, just the sound of the pump upsets her teenage son, and he doesn't like to be around when it's switched on. So it really shows you the variety of challenges to lymphedema patients. Patient support groups and LE&RN is the prime example here. Is hugely important for advocating for better solutions and better care for lymphedema patients. There's an important post-cancer care charity here in the UK called Macmillan that has made this estimate that if for every pound we spend on preventing lymphedema, it saves about 100 pounds eventually in reduced hospital admissions associated with the lymphedema. And then finally, surgeons. Well, there's unfortunately a variety of perspectives in the surgical community, and we're only showing two here. So, often you'll hear surgeons claim without any evidence that lymphedema is going away, due to changes in surgical techniques. And while that may be true to some degree, there's actually not much proof of it. And so whenever I hear surgeons say that, I always challenge them to provide the proof for that, and rarely can they do it. But there are some patients, and we work with some who care deeply for the possibility about the possibility of their patients getting lymphedema, and do try and take measures not only to do the right thing in surgery to minimize the risk, but also to inform their patients about lymphedema. So this whole question about how many patients get it, what's the incidence rate, is a very important aspect for someone who does research on this, like myself. So our question for the next couple of slides is, what is the incidence rate of BCRL? Most of us in the research community rely on a paper that's about 10 years old now, which in the research world is fairly old. And so what they did was summarize the results of 72 papers and compile them in a very carefully controlled statistical way. And when this is done in this way, it's called a meta-analysis. So they did this meta-analysis of 72 papers from the year 2000 to 2012. They found an overall incidence rate of 16.6% when they just compile all of the results of all those studies together. When they looked at prospective studies, in other words, studies that started out without knowing who was going to get lymphedema, so they pick a population of patients and they just watch them prospectively over time. The incidence rate was about 21.4%. with a 95% confidence interval, as you can see here, about 15 to 30%. So a lot of uncertainty in this, but you can see now why when we talk about the incidence of lymphedema, we say it's generally around 20%. And that's the accepted number in the literature. But again, some breast cancer surgeons will cast doubt on that number. So they'll say, it's only about 5%. There's not really any evidence for that. Now, since this paper is about 10 years old, my group and I, a couple of years ago, started in the very laborious process of trying to do an updated meta-analysis. And I'm pleased to share with you today some very, very preliminary results. So don't take these as gospel truth, but just wanted to share with you some insights that we've learned. So this plot here, and I'll just start with the horizontal axis here. This is lymphedema incidence. When we take all of the studies that up to date, we find an overall incidence rate of 14.9%, which is sort of equivalent in the way it was measured to this Discipio number of 16.6%. And you can see based on the confidence intervals here expressed in gray, that really these two numbers are not statistically significantly different. So the incidence rate hasn't really come down based on the papers that we've studied. Now, what our analysis, because it's more updated, it goes through 2020, and we're just in the process of updating this to 2022, is and we've got 183 papers compared to the 72 that were available at the time Discipio did their work. And the other thing that we've tried to do is determine which patients are at highest risk of contracting lymphedema. So what we have here are patients that have undergone sentinel lymph node biopsy plus surgery, plus non-surgical treatments, and that would be chemotherapy, radiotherapy, hormotherapy. And then patients that had sentinel lymph node biopsy plus both of those. Up here we have axillary lymph node dissection. So that's more complete clearance of several lymph nodes from the axilla. And so that's plus surgical treatments plus non-surgical treatments in the combination of the two. And you can see the incidence rate for sentinel lymph node biopsy by itself is around 5%. And generally, most of these studies show incidence rates of below 10%. But then once you get up into AL&D, then the incidence rates go up. Now, and you can see the number of studies and the number of patients. So this number is based on 30,000 patients, right? So there's a lot of data included in this study. Now, one of the challenges that we had actually in doing this is that reporting practices vary a lot amongst these 183 papers. So, for example, in a paper that describes AL&D plus surgical approach, you can see the incidence rate here in kind of the low 20%. They will tell you that they had these two treatments, but they will not tell you if they had non-surgical treatments. In other words, there will be some of those patients in these studies that had chemotherapy, radiotherapy, hormotherapy, and there'll be some that didn't. And they just did not distinguish that in those papers. So that creates a lot of uncertainties because we really don't know how to distinguish between these subsets. And if we had better reporting in the literature, we'd actually stand a lot better chance of telling these patients apart. So that's one problem, is just the lack of good detailed reporting in the literature. Another aspect of trying to figure out what the incidence rate is, and I'll remind you these are really preliminary results, is the diagnostic technique. So you can see if you look at arm circumference, if there's an increase in arm circumference greater than 10%, if that's your criterion, then the incidence rate for all of those studies is about 10%. But if you change your criterion to be greater than a 5% increase in arm circumference, then you get an incidence rate of about 24%. And if you look at just raw increase in arm circumference, if you say it has to be at least 5 centimeters, it's here. And and you can see that goes up to 20%, down from 10% up to 20%. So these incidence rates are doubling based on just variations in diagnostic criteria. And here, this is volume of the arm. And you can see if you say it has to be at least 10% increase in volume of the arm, the incidence rate's about 16%. And it more or less doubles if you say that 5% and greater is the criterion. So incidence depends highly on the diagnostic method, and all of that is mixed into the incidence numbers that I showed you on the previous slide. And that is a big, big problem in trying to figure out exactly what the incidence rate is, not only overall, but for all those treatment combinations that we talked about in the previous slide. ladies and gentlemen, we need standards. We need a better way of reporting all of these clinical trial results. And all of these 183 studies that we looked at, they all do reporting in different ways, and it's not great when you're trying to figure out what the incidence rate is. So, what is the incidence rate of BCRL? Having studied it for a few years in this meta-analysis, honestly, I know less now than I did before. Right, it's still about 20%, and I will keep saying it's about 20% because that's what the evidence that's out there. But if you ask me for an exact number, I have to admit I have no idea. So there you go. But I will tell you this: one thing that we've learned from our stakeholder engagement is that our patients tell us they don't really care what the incidence is. Very few of them were told about what the incidence rate is before their surgery. And Stan Roxon showed us some interesting data when he was visiting a few weeks ago about what patients are told, and it's shocking. And I look forward to that publication being out there. But if incidence rate is not even being shared with the patients, then what good is it? So I'd like at this point to introduce what we've adopted as our approach to motivating the seriousness of breast cancer-related lymphedema for the sake of our research proposals when we're pursuing funding for the basic research as well as the more applied stuff you're going to hear about today. And that's the engineering concept that risk is the product of likelihood times severity. So if something is really not very likely, it's it's really like getting struck by lightning. Well, the risk is just not very high, even though severity might be quite bad. And the airline industry has actually taken this approach to determining risk. So obviously, if you're designing a passenger airplane, the severity of a malfunction is very, very high. So what you try and do is just lower the likelihood. And the airline industry has actually been very successful at that. It's very rare that you hear about bad things happening in passenger airplanes anymore because they've used engineering techniques to identify potential issues and deal with those. And so in that sense, we're looking at a similar problem here for breast cancer-related lymphedema. The severity is measured physically, emotionally, economically, and these are all bad numbers, and they're not likely to change unless we find a way to reduce the likelihood. So, like the airline industry, that's what we've decided to do. We're going to work on the likelihood of breast cancer-related lymphedema forming. So, how do we engineer a solution? Well, in my lab, we study not only the biology of the lymphatic system, but we study the pumping mechanics, we study fluid flow. So I actually trained as a mechanical engineer in my undergrad days. And so I learned a lot about fluid flow back then, and I really enjoy applying that to the lymphatic system. Fluid mechanically, it's an absolutely fascinating system. So as I said before, we do some experiments, we do some mathematical modeling, and most of all, we pay attention to what the system teaches us. We stare at our piles of results from our experiments or our computational models for a long time. And we think about what they're telling us, and then we think about what we can do to help solve the problems associated with the lymphatic system. So I'm going to run through just a couple of examples of the background research that led to the development that you're going to talk about today. So the first is to explain a little bit about lymphatic fluid flow. So this is a movie of a rat mesenteric lymphatic vessel. So you can open up the rat, stretch out its mesentery over a microscope stage, and easily find a lymphatic vessel. So you can see the lymphatic vessel here. It's contracting, it's relaxing. You can see the flow going through it. So those are immune cells being delivered to a lymph node. And you can see they speed up, they slow down, they slosh back and forth a little bit. And so this is fascinating to a fluid mechanician like myself. So we can measure the velocities of those cells. We can measure the movement of the wall. And what we find is that these cells are moving at about 1 centimeter a second, which is pretty fast for flow in such a tiny vessel. And the flow rate in this vessel, and I love these units. If you're an engineer, you'll appreciate these units. 200 microliters per hour. So if you can imagine how much a microliter is, and there'll be 200 of them moving through this vessel in an hour. This vessel is about 100 microns in diameter. That is about the diameter of a human hair. Now, what's keeping these the cells moving mostly forward is just out of the picture on either side here is a lymphatic valve. So these are bileaflet valves, and they shut when flow tries to move back in the wrong direction. So, so that's how lymphatic vessels pump, is with the combination of squeezing and check valves. And so lymphatic vessels do what all pumps do. They take fluid from a low height and move it to a higher height with the input of energy and the flow of the contraction of the vessels. And then the flow moves in this direction here. And how much flow you can generate depends on the pressure difference that you have to overcome here. So that's your basic fluid mechanics lesson for the day. So then what happens is this fluid comes in from these vessels up here in the top right and the bottom right, and they come into a lymph node. Now, when those lymphatic vessels are pumping, remember, they're about the size of a human hair, maybe a little bit larger, and they can not generate very much pressure. So if they're presented with a lot of pressure downstream or a lot of resistance downstream, it really impedes their pumping a lot. Now, what nature has figured out, which is why the title of the seminar is Nature-Inspired Solutions. What nature has figured out is that if it splits the lymphatic vessel into multiple parallel pathways, and you can see in this case, this vessel has split into 3 parallel pathways. So it has this one down here. Then what that does is it minimizes the resistance that the lymph node presents to that upstream pumping vessel. Now, lymph nodes, I can tell you, are a couple of centimeters across in humans. They're packed with millions of immune cells, like a very, very tightly packed sponge. And they actually do present a lot of resistance to lymph flow. And without this strategy of splitting into multiple parallel vessels, a single vessel would not be able to pump fluid swiftly enough through the lymph node to deliver the immune information, keep fluid balance going, and all the important things. So you got these multiple parallel vessels coming in to the lymph node, and you typically have one vessel leading out from the lymph node. So that's the that sort of feature of lymph node fluid flow caught our eye, and we decided, all right, well, if we're going to come up with something that can keep fluid moving like a lymph node, we need to split flow into multiple parallel pathways. So this is the solution we came up with. We call it Faciliflow, and it's designed to restore lymphatic vessels following surgical removal of a lymph node. So you can see it has 6 parallel pathways here for fluid flow to go through it, and 2 suture tabs on the end here to secure it in place. And I'll explain how it goes in the next slide. But you can see it's a few millimeters across compared to a normal green pea, garden pea. You can see how big it is. And so the idea is that the surgeon would put this in after they remove the lymph node. Any surgeon can do this. They don't need specialized skills. They just need to be able to tie a knot. All surgeons can tie suture knots. It's part of the basic training. And then we go in after the lymph node has been removed. So the idea is that the surgeon removes the lymph node that leaves these vessels hanging sort of in the tissue around them. The implant goes in place of the lymph node that was removed, and hopefully the vessels reconnect. So not only is the vessel reconnection encouraged by the minimization of flow resistance with these parallel channels here, but we're also encapsulating inside the implant VEGF-C. So this is vascular endothelial growth factor C, which is more or less specific to the lymphatic vessels and will encourage those vessels to regrow. So we're not applying VEGF-C systemically throughout the whole body. We are applying it only in a local region right around where the lymph node was removed, and it shouldn't have any effect outside the immediate region of the implant. So when we came up with this idea, we thought, all right, well, we need to test it in a simple, low-cost, easy animal model. So we did this in rats. We found some other results in the scientific literature showing the effect, how effective it is to do autologous lymph node transfer in rats. So that's autologous lymph node transfer, or ALNT, is a surgical treatment where they take a lymph node from another part of the body and move it to the place where the lymph node has been removed. So the rat version of that procedure, when you do that and you administer VEGF-C along with the procedure, you find that you can reconnect just over half the vessel. So 53% of the lymph node fragments that they put in place of the removed lymph node will show evidence that the vessel grew back around that lymph node fragment. When we put an early version of our facilitated implant inside these same rats, we found that we had a reconnection rate of about 55%. And again, that's with applying VEGF-C in solution at the time of surgery. So it works at least as well as something that's used surgically now to try and deal with lymphedema. And so that gave us a good early indication of potential success, and enabled us to get some funding to pursue this idea further. Now, this is where the real engineering work begins. It's not an easy thing to do, to come up with an implantable device, and this would be a permanent implant, stay in the body forever. So you need to address safety, manufacturability, sterility, and testing. So a lot of good engineering goes into this, and I'm going to just sort of expose you to some of it in the next couple of slides here. So in terms of safety, we are using materials and a drug release concept known to be safe since the 1980s. So there are lots of permanent implants in people's bodies all over the planet using the same material that we use, which is silicone. We are using albumin as a way of releasing the drug, and the drug is VEGF-C. Releasing drugs from permanent silicone implants using albumin is something, again, it's been around since the 1980s. It's used in birth control devices to prevent infections, lots of different chemotherapy devices used in a lot of different areas of medicine. So this is very, very safe, we feel, and we hope that when we get to clinical trials, that the regulatory bodies will accept our claim that it should be very safe. Manufacturability, where we're using a technique called injection molding. It's very commonly used in many areas of industry, and to combine all of these materials together, mix them up in something the size of a garden pea, and injection mold it will cost less than about $10 when we get to manufacturing. Sterility is something that we've been investigating, and then you need a way of testing the implants as they're coming off the assembly line. You pull one at random, you test it, and make sure it does the things that you expect it to do. So in terms of designing the release mechanism, again, we're combining silicone with VEGF-C and albumin. Albumin is a water-soluble protein. It's the most common protein in the body, and very common, very safe. And so when you mix the silicone with the albumin and the VEGF-C, and the albumin starts to dissolve in the water inside the body, it creates these channels inside the silicone and releases the VEGF-C gradually over time. So you can see the release kinetics here. So this is the number of nanograms that's been released, and the percent of the VEGF-C, in this case, it's a dextran surrogate, the percent that's been released. So we've released almost all of the drug from the implant by about 100 days, which we think is a good target. We want to provide the drug in sufficient concentrations for about 90 or 100 days to ensure the regrowth of the lymphatic vessels. And you can see if we don't include the albumin, We only release what's at the surface very early on. So all this release later is due to the formation of these channels as the albumin dissolves and the water gets in and pulls the VEGF-C out. We can witness this in real time. So there's an implant in the middle here with a fluorescently labeled drug surrogate. And when we can watch that using a microscope as it leaks out, it shows up as this green color here. And we can measure that over time to determine how far it goes out into the tissue, how fast it gets there, and how long it lasts. So that's all part of the engineering of designing the release mechanism. Now we can do some computational modeling as well. So here is the basics of the computational model. You see the implant here in the middle. There is interstitial fluid flow going through here. And what you'll see is that there's an initial burst of VEGF-C release, that big cloud that comes out here and quickly goes downstream. So that is the only VEGF-C that would be released if we didn't have the ability to release the drug from the inside of the implant. And then we can plot things like, so 2 centimeters away from the implant, how does the concentration vary in time, depending on how much VEGF-C we have inside the implant. You can see the more you put in, more comes out. It sort of makes sense. Now, the therapeutic level of VEGF-C is about 100 nanograms per milliliter, so about this level here. So we would need, in order to make sure that there's enough VEGF-C at 90 days, we would need to load it up with about 60 micrograms of VEGF-C. So it gives us a good idea how much we need to load in there. Now, the other thing we can do is vary the amount of albumin and the mixture and the way we mix it all together to vary the diffusivity of VEGF-C in the implant. So how fast is it released from the implant? And you can see if it's not released very fastly, not much of the drug comes out. If you, at the intermediate diffusivity here, you can see that the concentration stays right around the therapeutic level, so that's good. But if you have, if the diffusivity is too high, then these initial concentrations are probably too high. You probably ought to back off on that. So we've been looking for something in between here. So these computational models, you can see, tell us about a lot about how we need to design this implant in order to achieve our medical goals. And then we need to be able to test the implants. So the way we do that is we construct a cell culture experiment. So we take lymphatic endothelial cells. These are the cells that line the insides of your lymphatic vessels. And we culture them inside the dish, and then we scratch across the layer, creating a wound here or a gap where there are no endothelial cells. We drop an implant into this solution here, and then we watch how fast that wound closes over time. So the wound healing rate is not very high if you don't have any VEGF-C in there. You put VEGF-C in there, it gets higher. And then we can use this to look at a couple of different sterilization techniques. So this is ethylene oxide gas as one sterilization technique, and gamma irradiation is another sterilization technique. And you can see that the gamma works a bit better. So that's the one we're moving ahead with. And I'll say this is also using this sort of scratch test assay here, as it's called, is going to be a valuable tool for us as we start manufacturing these things. And again, we can pull one randomly off the assembly line, drop it into this experiment, and make sure that our manufacturing processes are really producing exactly what we intend. So we've also done a bit of business planning around this device. So you can see in terms of the competition here, we are a preventative technique. Lympha is a prevention technique. It is the version of LVA that's done at the time of cancer surgery. So LVA is lymphovenous anastomosis. It's where the surgeon takes the lymphatic vessel and connects it to a nearby vein. ALNT is autologous lymph node transfer. That's where they move a lymph node from one part of the body to the other. I described that to you earlier. These all take a couple of hours. Both of these surgical procedures take a couple of hours. This one's a bit shorter because you already have the patient open and you're doing the breast cancer surgery. You can see they're all pretty costly, in the $10,000 range and upwards. And these would be, treatment that would be additional separate surgical procedures. So, these, because they require specialist microsurgeon skills, are really not available to all patients out there. So we want our device to be able to be deployed in any breast cancer surgery. And again, any surgeon can tie knots, and that's all you need to do to put this device in. So it goes in during the cancer surgery, very simple procedure, minimal intrusion on the standard of care. We think it would take at most 5 or 6 minutes to deploy this device. It's very low cost. We think we're going to end up charging about $1,000 or £1,000 for the device, and that's comparable to other medical devices of similar complexity. And we really hope this will be available to all breast cancer patients everywhere. So in terms of where we are on a timeline, well, we've got the patents issued in several geographies around the world. We've been doing R&D and design updates on the device itself, and you've seen some of that in the past few slides. We'll be setting up our supply chains for VEGF-C and medical grade silicone, and doing more animal studies in the next couple of years. And we're sort of writing funding proposals like crazy for that. And then hopefully by the end of this decade, we'd be into clinical trials and seeking regulatory approval for the device. When you're putting in a preventative device like this, you have to really address safety in a very serious way, and we think we're doing that. In terms of the market, well, it's a fairly big problem. The market for this device we anticipate to be in the sort of hundreds of millions per year in terms of total addressable market. And you can see China actually represents a big market for us. We have patent protection there, and so we hope to be pursuing that market. It's a hugely growing problem in China. In terms of healthcare economics, we know it's very expensive to take care of breast cancer-related lymphedema patients. So the cost of taking care of a BCRL patient for 5 years in the UK is about £36,000. In the USA, it's about $26,000, and again, that's spread over 5 years. And in the EU, a bit less, but this is tens of thousands per year for the 5-year period for the rest of that patient's life, and survival rates are in the range of 80 to 90% for breast cancer. So we're going to be paying for these lymphedema patients for a very long time, and that compares to our one-time cost of about $1,000 to get the device in there. So if this succeeds in preventing lymphedema, it saves all this money, and so the health economic case is quite strong for this. So this is our team. I'm very pleased to introduce them to you. So this is Arushi. She's a postdoc in my group and an R&D engineer. She came to me with medical device design experience, and she's just done a wonderful job in preparing this device, along with Alain Vella, who came from, again, experience in medical device development at a large multinational company you've probably heard of. Kate Rowley is our CEO. She's been a part of many biotech and medical device efforts as both an investor and in charge of those companies as well. We have a wonderful breast cancer surgeon that helps us out named Paul Thiruchelvam. I'll point out this Mr. here. That's the standard in the UK. He is actually a doctor, but we call surgeons Mr. For historical reasons. And then these are the hands of 3 breast cancer-related lymphedema patients who advise us at every stage along our research and development project. They are actually holding the implant in their hands there, and you can see the compression garments that they're wearing. I got to tell you, we would not be anywhere close to where we are now without these patients. They're super brave for helping us with our research and coming out and talking about their condition. And we can't thank them enough for their involvement in our research. Our engagement strategy, and now really the point of this slide is it's complex, right? So don't worry if you don't get all the aspects here. So we would supply medtech distributors with our implant. They would interface with the healthcare providers who need to hear from the surgeons that they want the device. Healthcare regulators are hugely important. They determine whether or not we can sell these things, and they provide feedback to all these other stakeholders. We need the patients involved, we need the surgeons involved, we need policymakers, we need the insurance companies, we need payers involved, and all of that ecosystem is required for success. And again, we've designed our device so that it fits most easily within this very complex healthcare environment. So in summary, so I've told you a bit about our research and about this device that we're developing. And here's a nice piece of artwork that shows you the concept. So again, the surgeon would take out the lymph node, leaving the vessels behind. Within a few minutes, the implant can be in place, releasing VEGF-C, providing minimal resistance to fluid flow, hopefully ending up with vessel regrowth on the order of, say, weeks or a few months after the surgery. And hopefully we prevent the breast cancer-related lymphedema from ever forming. We're still under development. Our design goals are to be simple to integrate into the standard of care. Safety is hugely important. We're giving this device, it's kind of like a vaccine. We're giving this device to someone who doesn't have the disease. So it needs to be as safe as a vaccine in that regard, and of course, as effective as it can be. So there's lots of engineering, lots of biology, business planning, stakeholder engagement, et cetera, involved in the development of this project. This is true for all medical device technologies, and but it's a lot of work. It's hard work, but the possibility of having an impact on patients' lives in this way is so exciting and would be wonderful. And so that's what drives us every day as we come into work and pursue this. And we need a fair amount of funding as well. So like I said, we're writing proposals like crazy to get this thing to the next stages. And hopefully we'll get it, keep it moving forward. So with that, I will stop sharing and hopefully be able to answer some questions. Okay, so first of all, Hari Shankar, thank you for your question. Could this device help for primary lymphedema in the leg? So with most cases of primary lymphedema, that is occurring either due to lack of pumping or malformation of the valves, those bileaflet valves that I showed in the leg. I'm not sure there's much this particular device can do, because those patients have lymphatic vessels. They don't necessarily need more lymphatic vessels, but if it turns out that that would help them, then that's something we would like to do. But I'm not so sure it's going to help. So Cherie asks, what happens in the severed lymph vessels after surgeons remove our lymph nodes? Well, from what we've learned of the surgeons that we've talked to, it appears that the lymphatic vessels are still there, but when they're cut, they retract into the tissue by a centimeter or two, according to the estimates of these surgeons. And that's why we want to provide that therapeutic concentration of VEGF-C at a distance of 2 centimeters away from the implant, is to reach those severed lymphatic vessels. So they're still there. There's some evidence that they might wither away with time, but that would be over weeks to months. And again, what we hope to do is prevent that by encouraging them to regrow. So as one thanks for the presentation, you're welcome. It's my pleasure. Is the device affected by radiation therapy? That's not something that we've looked at yet. It would be surprising to us if it did. We actually use radiation to sterilize the device, although that's a different kind of radiation than what gets used on patients. So we will take that question and try and answer. Of course, the real efficacy of the device is not going to be known until we get into the clinical trial. So it may be later that we find out the answer to that question. So Euripides asks, is the number of lymph collectors going in the lymph node the same as the number going out? then if not, how can they fit in the small device? So the holes through the device are about 1 millimeter in diameter, which is a reasonable, slight overestimate of the diameters of the typical lymphatic vessels in the arm at that point where they're approaching the lymph nodes in the axilla. So we think a millimeter hole gives them plenty of room to grow through there. So we expect that there would be multiple incoming lymphatic vessels, maybe 5 or 6, and typically only one lymphatic vessel going out. So if there's, if we manage to regrow any lymphatic vessels, I think we're happy because that keeps the fluid moving, and the more lymphatic vessels, the better. So we're not really trying to fine tune this down to the point where we're imitating, 6 vessels in, 1 vessel out. We're just trying to regrow as many vessels as we can. Right. And another question, it was mentioned that vessels will hopefully reconnect. Does the device work as a connector? Well, no, it really just sort of relies on the vessels doing the reconnecting to each other with the encouragement of the fluid flow and the VEGF-C together. And by the way, the combination of fluid flow and VEGF-C works better than VEGF-C alone. So we're hoping that plays in our favor as well. Cheryl asks, I had 47 lymph nodes removed. Would the surgeon have to replace a silicone faciliflow for all 47 or only a portion? This is an issue that we've thought about a lot. There's not a lot of evidence out there yet because we've not deployed this in large animals yet. We hope to have some answer to that as we go along in our research and development process. I would say, given that we aim to regrow vessels within a couple of centimeters of the implant, I would say if the 47 lymph nodes were fairly close to each other, we would not need 47 implants. We would, if the lymph nodes are being removed within a sort of 3 or 4 centimeter neighborhood, I would only put 1 implant going in. We don't have much factual basis for answering that question, but we don't think it's going to be 1 for 1, particularly in cases where the original lymph nodes were close together. So Greg King, hi Greg, glad you were in the talk today. Greg asked, excellent presentation, thank you. The preliminary data you showed, 14.9% incidence rate, is this taking into account the surgical volume of SL&B versus AL&D performed? That's all of them, Greg. So that's all of the SL&B patients and all of the AL&D patients and studies all lumped together. Are these AL&D surgical cases decreasing? That's something that we need to find out because the data that we're basing our meta-analysis on, that requires other investigators to do studies of SL&B or AL&D and these different combinations of surgical and non-surgical treatments. And the number of patients and the percentage of patients that get SL&B versus AL&D in the literature of the studies is not necessarily representative of the whole population of breast cancer patients. So we don't have an answer to your last question, is AL&D decreasing, but we need to know what's happening with the proportion of patients getting SL&B versus AL&D in order to interpret our results. So if anybody out there has some good information on that, I'd appreciate hearing about it. We're just starting to dig into the literature on that ourselves. We have an anonymous question. Will you discuss head and neck lymphedema help at all? It's a serious problem, obviously, for head and neck lymphedema. The incidence rates are very high for that type of cancer, and we hope we can help those patients. We hope we can deliver the device into those markets as well, but we'll be pursuing the breast cancer indication first, simply because it's the largest one. But I know that head and neck is very serious, and we hope to get there. And as we go along, we may change strategies and decide to address that one first because it is so prevalent. So thanks for your question. Sally Waterman asked, "Have you any data showing the release kinetics for VEGF-C in addition to dextran?" Yeah, so we've done both the VEGF-C and the dextran. The reason I showed you dextran today is that dextran is cheap. It's basically just a natural sugar, and it comes fluorescently labeled. So that green shadow you saw in that microscopy image there is a fluorescent image of the dextran. And it's very difficult to label VEGF-C with a fluorescent tag, and if you do, you probably change its function. So we're just not, we've got other ways we can measure VEGF-C concentrations, but we can't image the spatial variation in concentration like we can with the dextran. We've done what we can to make sure that the dextran has similar properties to the VEGF-C, and actually we're using another compound now that behaves more closely to VEGF-C and not dextran. So thanks for your question. Adrienne asked, how will you determine who would be a good candidate for Faciliflow? So that was sort of the goal of our meta-analysis, where we were looking at different surgical combinations. So when we went for peer review of our previous research grant, one of the reviewers said, well, you should find out which subset of breast cancer patients is most likely to develop lymphedema so that you can address them first with your implant. And I guess there's some logic in that. I could argue against it, but if a reviewer says it, we have to do it. So that was what we were looking for in that study. And you could see there was, on average, the incidence rate was higher for patients that get, the AL&D plus the surgery plus the non-surgical treatments. But again, just because of the poor reporting practices in the literature, it didn't come out to be statistically significant. So we think we probably go for the AL&D patients first over the SL&B. I mean, still 5 to 10% incidence of lymphedema as we're seeing with SL&B is not negligible. And again, our patients tell us they don't really care what the incidence rate is. They care about the risk, which is the likelihood and the severity combined together. So we think we can help a lot of patients here, especially if this ends up being as safe as we expect it to be and as successful as we hope it can be, then I think it should be available to all patients. So thanks for your question. Cynthia asks, is your device more effective than the LVA or the ALNT? We don't know that in terms of humans yet. We have the evidence in rats that it is as successful as the rat version of ALNT, which is not the same ALNT as they do in humans. And we don't know that yet. We expect that it will be, but we don't know yet. Bonnie Scheidler asks, if you have multiple lymph nodes removed, do you only put in one facility? This is similar to the previous question. So yeah, if the lymph nodes that were removed are close by each other, then I would not do one for one. But that remains to be seen what that strategy needs to be. And yeah, only 6 minutes to perform. And I think it should be less than that, actually, because all this patient's got to do is put the implant in place of the original lymph node, orient it correctly, and tie a couple of sutures. And if you've ever seen surgeons tie sutures, you know it only takes some seconds to do that. So I think 6 minutes is probably an overestimate of the amount of time necessary to deploy this implant. So thanks for your question. Sally Waterman asks, is the 90-day release profile based on estimated time for vessel reconnection to the device? And if so, it's just based on data from rat or man. That is based on a number of studies in the literature, the sort of time course of lymphatic vessel regrowth in mice and in rats, and to some degree in larger animals such as pigs and humans. It appears to happen over the course of a few months. And so it's a rough estimate, but we think it's as good an estimate as we can have. at this point that we want to provide that therapeutic drug concentration for a period of 90 days. So we hope to find out. It'd be nice to know that more exactly, but that there's not really any more information out there in the literature. So let's scroll down and get some other. Everybody, thanks for all the questions. Maybe you can email me, and and any of you that I don't get to your question, I'll be happy to answer questions by email, which was my email address was in the slides. So, or you can just find me through Imperial College. So, which countries will you be seeking regulatory approval? So we have patent protection in the US, the EU, and China. And so those would be our initial target markets, and then hopefully spread it out from there. So, Mark, would this device have applicability in other lymphedema models? Not necessarily primary lymphedema, but I think any surgical procedure where lymph nodes are being removed for whatever reason, I think we stand a good chance of addressing those applications. Primary lymphedema, again, that's usually due to problems with vessel contractility, which affects the pumping and also the formation of the valves. So that's not something that this implant could really help with, unfortunately, in its current design. We thought about putting valves on the inside of it, in which case maybe it would help, but that's something for down the road. Does the device need to be swapped out after a certain period of time? That's not the idea with this one. It would stay in there forever. Again, there have been silicone implants that release drugs going into humans since the 1980s, and they stay in there forever. So a lot of them do. So that's not going to be a safety concern that it stays in there. We also considered the possibility of making the implant biodegradable, like some implants are. That comes with its own set of problems. So you got to decide how fast you want it to degrade. What's going to happen as the device degrades? How is that going to affect device performance? And then when these biodegradable polymers start to go out into the body, they can change the acidity in the surrounding tissue, and that can create inflammation. That's probably going to affect the lymphatic vessel regrowth. It might benefit the regrowth. It might be bad for it. We don't know, and we'd rather not start asking such complex questions at this stage. So maybe version 4.0 of the device will be a biodegradable version, but we think keeping it in there forever is going to be safe enough. Then that's what we're going to go with for now. All right, thanks everybody for dialing in. Hopefully you got my email address in the presentation, but you can track me down through Imperial College. And thank you for your attention today. Thank you for your support. Thanks for all the great questions. I wish I'd gotten to them all. There were 68 questions online, and that's impressive. That's wonderful. It's a lot more than my students ask when I lecture here, so I appreciate that. And have a good rest of your day there. Thank you very much. Bye-bye.